This is a video on how mutations fit into the gene expression achievement standard. So I've written some keynotes, which you may like to copy. Okay, so there are three main types of point mutations that are involved in the gene expression standard. So the three types are substitution, insertion, and deletion. So they are the ones in the red. So we'll take substitution by itself first. Substitution is generally thought of as not as serious a mutation than insertion and deletion. So, substitution mutation is when one base has been swapped for another base. So C has been taken out of the DNA and A has been put in. So mutations only happen to the DNA strand. They're not happening to the mRNA strand. Let's keep it simple anyway. Whereas for insertion and deletion, that's when a base is either put into the gene, so that's insertion. So instead of having 20 bases in the gene, it will have 21. Or deletion is when a base is taken out. So instead of having 20 bases in the gene, there are only 19 bases in the gene's information. So they have different effects. So effects of substitution. Number one, it might cause a silent mutation. And the substitution mutation to the third triplet. So a triplet is a series of 3 bases on the DNA. Every 3 bases code for one specific amino acid. So substitution mutation to the third triplet may not actually change the amino acid because of degeneracy. Therefore, the length and order of the bases is the same as it was in the normal order, and the final protein functions correctly. So degeneracy is when many codons code for the same amino acid. So if the third base is changed, generally the same amino acid happens. Missense mutation. If the substitution mutation occurs in the first or second base of the triplet, it's more likely that a different amino acid will be used in the protein. And so a missense mutation is when a different amino acid is created. Changing the amino acid sequence may change the folding of the peptide chain by changing all the bonding that happens between the amino acids. So it may not be able to carry out the same role as the intended protein. For example, if we think of enzymes and their active sites, how they've got really specific active site shape to fit only one type of substrate. If there's a different amino acid in the mix, then the properties of the folding may be different. So you may not get the same active site shape. And if you don't get the same active site shape, then that reaction won't happen because they won't fit together like a lock and key. It'll be like having a different lock and the key can't fit. So that reaction won't happen, and that can actually be really dangerous for the body. Okay, so the third effect of substitution is a nonsense mutation. And that's when the produced triplet, so the new triplet, will produce a stop codon. This can have a large effect since translation would stop too soon and would have a short non-functional protein. So if the mutation happens right at the beginning and it creates a stop codon, then none of the subsequent amino acids will form. They won't join together. So instead of being this long, the protein will be this long. And of course, then it won't fold into the right shape. So that's bad as well. Okay, insertion and deletion. So when a base, so remember that insertion is when a base gets put in, deletion is when a base gets pulled out. So the effects of insertion and deletion, frame shift. So this is a biggie. By adding or removing a single base, every triplet from the point of the mutation is affected. Therefore, from that point onwards, they create different codons on the mRNA. So the reading of the codons on the mRNA after the mutation will code for different amino acids. This means that the final protein will be non-functional. It won't work because the entire amino acid sequence from where the mutation is onwards is incorrect, therefore causing the folding of the final protein to be abnormal and non-functioning. This will produce a completely different protein from what was intended. So it may produce, there's two things to that. So the intended protein won't be produced. So if it's an enzyme that's needed for a reaction, that reaction won't happen. And a different protein will be created, and that protein could actually be dangerous for the body. Another possible effect of insertion and deletion is a nonsense mutation. And the frame shift could alter the triplets that create new stop codons, which affect the length of the chain and the structure of the protein. So same deal as the substitution. When there's nonsense mutation, stop codons created, therefore there'll be no translation after that. So instead of being this long, the amino acid chain will be this long, so it will not fold in the same way. Hopefully that was handy. If you need a copy of the little cheat sheet here, talk to one of your teachers in the biology department and they will get that for you. So hopefully that was good.